Hello, I'm Sean Ekins, CEO of Collaborations Pharmaceuticals, and I'd like to briefly introduce our work on an enzyme replacement therapy for CLN1 Batten disease. Batten disease is an ultra-rare disease composed of over a dozen different forms, which all need their own treatments. We're working on just one, called CLN1, in which the children have progressive developmental motor, language deterioration, seizures, loss of vision, and early death. The disease is caused by mutations in the CLN1 gene, coding for the protein palmitoyl protein thioesterase 1, or PPT1, resulting in a reduction or absence of activity. Children may be diagnosed as early as 6 months of age, and we usually hear of about 2 to 3 children identified a year with the disease. It's thought there may be over 20 patients in the US, 10 in the UK, and over 20 in India. It's likely there are hundreds of children with CLN1 around the world, and many undiagnosed. There is currently no treatment, and CLN1 families have been very helpful in supporting our efforts to develop a treatment. The approach to delivering enzyme replacement therapy is not new. It has a proven track record and is recognized by the FDA. It also has safety advantages over gene therapy, and in other diseases can stabilize the disease outcomes, leading to improved quality of life. With an enzyme replacement therapy, there's also the potential for in utero treatment. There are, of course, challenges in getting the protein to the brain, which we overcome by using intracerebroventricular delivery, as used with Brineura for CLN2. The protein needs to be delivered frequently, likely every few weeks, and while these treatments are not cheap, they are a lifeline for families. Hypersensitivity reactions are rare because we're using an unmodified protein. There have been many years of testing the ERT, whether delivered IV, IT, or more recently ICV in the CLN1 knockout mice. Our most recent work, published with collaborators, indicated that ICV delivery normalized the gait, motor abnormalities, neuron loss, glial activation, and storage material accumulation in mice, all hallmarks of the disease. In order to bring this treatment to fruition, we're working with a number of groups, including STC Biologics to manufacture the protein. RTI International have helped us with the pre-IND and regulatory filings. Northern Biomedical Research are performing the IND-enabling toxicology study in non-human primates, while our preferred clinical site is with Dr. Emily de los Reyes at Nationwide Children's Hospital, and CTI have helped us in developing a clinical protocol. We also have our quality systems in place, aided by Long Scientific LLC. In order to find CLN1 patients and find out about their diagnosis and stage of disease, we have developed our own registry and obtained IRB approval. Recently, we've analyzed the data, currently for 23 patients, which showed some statistically significant differences between groups based on their age of diagnosis, e.g. the age of motor decline in late infantile was significantly lower than in juvenile forms of the disease. With the goal of moving to the clinic as rapidly as possible, we've already designed a natural history study with Dr. Emily de los Reyes, which we hope to initiate soon. We've also developed a clinical protocol and submitted a grant proposal on this to the NIH. We need to file a patent on our formulation and process, develop a USAN name for the product and logo, as well as file a trademark in future. In parallel, we're looking at additional indications and uses for the recombinant human protein, PPT1, as well as biomarker development. We're continuing our patient advocacy and outreach so we can add more families to the CLN1 registry, with the ultimate goal to start the natural history study as soon as possible, in preparation for an eventual clinical trial. We will present 2 posters on our work at the upcoming World Symposium as well. We're not aware of any substantial competition developing a treatment for CLN1 that is as advanced as we are currently. All the gene therapies have been shelved, which leaves a limited number of families who are able to obtain these treatments through compassionate use. Because the disease is small, we believe the target market in the US is likely around $20 million per year, and outside the US, the market size may be similar. We have both the FDA orphan designation and the pediatric rare disease designation for this disease. We'd likely price the enzyme replacement therapy similar to other ERTs, e.g., depending on the size of the patient, the price of Brineura is approximately $900,000 per year in the US. Our CLN1 project has been funded completely by the National Institute of Health, NINDS. This includes the preclinical work in mice through a phase 1 SBIR. The business plan was refined with funding to attend the ICORPS program. A diversity award supplement enabled us to do additional work on the protein, while our GLP manufacturing and tox study have been funded by a phase 2 SBIR. And finally, our GMP manufacturing, quality systems, clinical trial protocol, and IND submission are being funded by a CRP grant. Many collaborators have been or are currently involved in this project, which is being coordinated by Dr. Renuka Rahman. We're grateful for their efforts and the support of the CLN1 families. If you are interested in learning more about our work, please reach out to myself, Sean Ekins, or if you're interested in licensing this protein, please reach out to Mr. Aaron Oliver, our Business Development Manager. Thank you for your attention.